Hello everyone. Today I am going to describe the Megasporogenesis. If you are the student of NEET biology, NEET, okay, NEET biology for 2026 and standard 12. So we are continuing the topic as a Microsporogenesis. We have already discussed. In this class, we will study about Megasporogenesis. Mega sexual reproduction in flowering plant. So today topic is Megasporogenesis. As we know that carpel is the female reproductive part of the flower. So when I draw the diagram of one carpel, so it has a stigma, a style, and ovary. This one is, and this one is septicus, oblique, कोई भी आप नाम दे सकते हो. तो ये फीमेल रिप्रोडक्टिव पार्ट हो गया. Megasporogenum, that is, it has further you can arrange the parts as this one is the ovule. Ovule-like structures in Megasporogenum develop inside the ovary. So ovary is meant for development of seed. Okay. So how the seed develop? Ovule actually form the seed. So development of ovule, try to understand. This area is called nucellus area. Nucellus area, and some of the nucellus cell become special and behaves like megaspore mother cell. So nucellus cell which is diploid, as we know that this flower develops from the after passing the juvenile phase or vegetative phase of the plant, enter into the reproductive phase, then flowering takes place. So while flowering takes place, the carpels come out, and when carpels develop, one of the part of carpel above the receptacles, thalamus, opposite to the thalamus. So one cell becomes special and behave like, that is nucellus cell, behave like megaspore mother cell. Megaspore mother cell. Okay, and that gives rise to ovule. Ovule-like structure, which is surrounded by cell. One of the cell here, try to understand. One of the cell, this end is called chalaja end, and this end is called micropyle end. So near the chalaja end, one of the cell, nucellus cell, these are the nucellus cell. More cells are found, all are called nucellus cell. Which are diploid in nature because there is no reduction in division taking place. This developed from the zygote and embryo. So diploid is there. Megaspore mother cell, myocytes. This megaspore mother cell undergoes reduction. Okay. So for megaspore mother cell undergoes reduction division to form full linearly arranged megaspore cell. As you know that during reduction division, 4 daughter cells are produced. So these 4 daughter cells are arranged linearly. Now what happens? The 3 daughter cells degenerate. One remains functional. The functional megaspore, gradual distance. This functional megaspore undergoes further mitotic division. Reduction division once takes place. Then it undergoes mitosis. Thrice. How many times? Thrice. 3 bar. First, one of the megaspore mother cell. First, it will form 2 cell structure in first division. Second division, there will be 4 cell structure. And in third division, there will be altogether 8 cell structure will be produced. Okay? So 8 cell structures are rearranged inside the embryo sac. This structure is called embryo sac in such a way that 3 cells lie at the top and 3 cells lie at the bottom. Means top bottom we can say near the chalazal end and near the micropyle. Please. It is good. Live class is good. So keep quiet. So this one is the egg cell. Here this one is egg cell. And these 2 cells guards the egg cells are called synergids. Synergids. So these cells are called synergids. And these 3 cells together called antipodal cells. Antipodal cells. One cell from all sides migrates toward the central part and these 2 cell structure join together to form central nuclei or ultimately secondary nucleus. Secondary nucleus. Okay? So in this way, the megasporogen, that is the ovule. Inside the ovule, the megagametophile, that is embryo sac. Embryo sac, which is ultimately 8 nucleated 7 cell structure. When you count, how many cells are there inside the embryo sac? There are 3, 6, or these 2. 8 nucleated, but these 2 join together to form single cell. So ultimately, is counted 1 mole. So how many cells will be there? 3, 6, or 1, 7. Therefore, the embryo cell, which is 7 cell and 8 nucleated. Embryo sacs are 4. Okay. So, how can you explain if the question comes? So first of all, you have explained that in carpel there are stigma, style, and ovary. Inside the ovary, they develop ovule. Okay? Or in the ovule region, some cells here as a nucellus cells become special as a megaspore mother cell. Okay? That have prominent nucleus, dense cytoplasm. Rest cell help them, and this undergoes reduction division to form linearly arranged 4 megaspore cell. Megaspore mother cell, what heard that? Megaspore mother cell undergoes reduction division to form megaspore cells. So these are linearly arranged megaspore cells. Mother word is deleted, megaspore cells. Out of these 4 megaspore, 3 degenerates, 1 remains functional. Functional megaspore undergoes mitotic division thrice to form 8 cell structure, ultimately 8 nucleated and 8 cell structure. Out of these, they are rearranged in inside the ovule as form the embryo sac. So an embryo sac, which is present inside the ovule, contains 8 nucleus and 2 polar nuclei fuse together to form secondary nucleus. So 7 cell and 8 nucleated structure is produced. So in this way, we can easily explain how the megasporogenesis taking place and 7 cell and 8 nucleated embryo cell. This is also known as monosporic development of embryo cell. In maximum plant, most of the plant, you will observe the monosporic. Monosporic, there is a question in your NCERT book you see. Monosporic development of embryo cell. Development of embryo cell. What do you mean by this monosporic development of embryo cell? Your answer again begins from there. That megaspore mother cell, some of the nuclear cell behave like megaspore mother cell. This is also known as megaspore mother cells or biocytes, which undergoes reduction division to form linearly arranged 4 cells. These out of 4 cells 3 degenerate, unlike the microsporogenesis. In microsporogenesis means formation of pollen grain. All pollen tetrads developed into pollen grains, but here all are not developing into into 5 embryo cell. Only one develops into embryo cell. Hence, this development is called mono. One means monosporic development of embryo cell. In higher classes, you will study that diasporic is also there, tetrasporic is there, all 4 developed in some of the plant, but beyond your 2 So this one is called megasporic development of embryo cell. So in this way, a mature embryo cell is produced, which contains, when I draw the diagram, this one is suppose that embryo cell having one egg cell lies at the center. This one is egg cell, and these egg cells are guarded by 2 synergids like a bodyguard, and one filiform apparatus is also there which guide the pollen tube development, and there are 3 antipodal cells lies at the opposite side of the here. Which end is this one? Can you guess which end is this one? This end is Shalaja end, and this one is end is called micropyle. Micropyle, and this one is Shalaja. So near the micropyle, the pollen germination, the pollen tube brings the male gamete, 2 male gametes, one fused with A and another fused with already fused to secondary nucleus. Hence the fusion is called 2 secondary nucleus fused together and 1 male gamete fuses. Here the fusion is said to be triple fusion. The fusion is said to be triple, 3 nuclei are fusing together. And fertilization is 2 fertilization. Once male gamete fuses with egg, another fuses with secondary nucleus. So fertilization is double. So what is double fertilization and triple fusion? And 7 cell, 8 nucleated embryo cell. These are antipodal cells. ये अलग से क्वेश्चन आता है आपका, आप पूछेगा कि व्हाट आर द प्राइमरीज ऑफ embryo cell. तो आप देखोगे कि ये ट्रिप्लॉइड 3n हो गया, ट्रिप्लॉइड हो गया. प्राइमरी इंडोस्पर्म न्यूक्लियस which form endosperm, that is a subnutritive value for the growing seed, that is embryo. वो embryo तो बनेगा ही नहीं, जायगोट फर्टिलाइज होकर के बनाएगा. और ये एंटीपोडल सब डिजेनरेट हो जाएंगे, डिजेनरेट सिनर्जिड्स और ओनली एक फर्टिलाइजर फॉर्म हो जाएगा एंड डेवलप इनटू एम्ब्रायो। तो इस तरह से आप आराम से इसका आप एक्सप्लेन कर सकते हो। तो इसका क्लोराइड क्लियर हो गया। ये जब फर्टिलाइज हो गया तो क्लियर हो जाएगा, फर्टिलाइजेशन के पहले है। और बाकी सब ये सिनर्जिड्स और एंटीपोडल सेल्स ये सब क्या है, क्लोराइड है। तो आपको हेप्लॉइड सेल है, एन सेल वाले भी हैं, डिप्लॉइड भी हैं और यहाँ ट्रिप्लॉइड भी है। तो व्हाट इज द क्लोराइड ऑफ एम्ब्रायो सेल? हेप्लॉइड, डिप्लॉइड एज वेल एज ट्रिप्लॉइड। ठीक है। तो आज मेगास्पोरोजेनेसिस आपको नेक्स्ट टाइम किया। थैंक यू।